Konnichiwa guys, welcome to our channel Loops of Biology. I'm Yogiraj Jakkal taking you on the next part of biomolecules. Now, the interesting part of biomolecules starts, and that is nothing but the application of biomolecules. What are the biomolecules we have learned before? So we have to take out proteins out of it and apply them inside the enzymes. So what is enzyme? Let's see. So what are enzymes? So enzymes, can you see this particular orange color ball? This ball-like structure is nothing but your enzyme. So there is going to be the enzymes are biocatalyst. They are nothing but your biocatalyst. Now what is catalyst? A substance or a molecule, a substance or molecule that speeds up the reaction, that is nothing but your catalyst. So this catalyst work inside a biological system. So here it is working in biological system, and that's why it is called as biocatalyst. Now whatever the chemical reactions which is taking place inside the biological system, that has to speeds up with the help of this enzyme. So the reactions become more specific and faster than usual whenever I am going to use this particular enzyme. Now in the case of enzyme, we have to learn one terminology that all enzymes are protein. All enzymes are proteins, but all proteins are not enzyme. Got my point? So here, if we try to draw a Venn diagram, so suppose if this is your case of protein, so this part is basically your protein. So out of the total protein portion, there is some portion inside a protein which is belonging to your enzyme. So all the enzymes are present in the category of protein, but the remaining part of protein is not acting as enzyme. So this part is basically no enzymatic action. Got my point? So whenever I'm considering enzyme, the enzymes are biocatalyst. They are made up of proteins, but all the proteins cannot act as enzyme. So the nature of enzyme is nothing but your proteinaceous in nature. What it is proteinaceous in nature. Got it? Now how this particular thing came from? Whenever I am considering the nature of enzyme, we have seen that the enzymes are made up of protein. The enzymes are made up of proteins. So when the scientist tried to isolate this particular enzyme, so whenever there was isolation of enzyme, isolation means taking out some of the enzymes from the biological system. So there was isolation of enzymes, and these enzymes were then studied for their structure. And can you see on the right hand side, there are all globular structures, right? So on they have seen many globular structure, many globular structure. Now, whenever I'm considering a globular structure, if we look into a detail, there has been a curly structure inside it, which is resembling here alpha helical structure, right? And these curly hairs are tied by a ribbon-like sheath. This is nothing but your beta pleated sheath. Got my point? So your proteins which are having your secondary structures are joined together to form a globular structure. So in detail, they have seen that the enzymes are made up of combination of combination of alpha and beta structures of proteins, and after that it has been clearly shown by the scientists and proved that all enzymes are nothing but your protein, but all proteins are not enzymes. So the nature of enzyme is nothing but protein in nature. Got it? So moving on to the next part, how do enzymes work? Whenever I'm saying the enzymes are reducing down a time for any chemical reactions. So whenever I'm considering enzymes, all right. So enzymes are doing, they are increasing the rate of reaction, right? The speed of reaction or the rate of reaction has been increased. Now, how will they increase? By decreases the time taken for a reaction, decreases the time taken for reaction. Okay. So now whenever I'm considering enzymes or any particular reaction, so the reaction to be spontaneous. So for spontaneous reaction, for spontaneous reaction, what do you mean by spontaneous reaction? That means it doesn't need any external energy and the reaction is taking place smoothly inside a system. Got my point? So whenever I'm considering a spontaneous reaction to take place, their Gibbs free energy has to be negative. So it should be less than 0. Got it? So whenever the Gibbs free energy is low, the reaction is going to be a spontaneous reaction. Got it? So in this particular case, the activation energy is going to play one role. Now, what do you mean by activation energy, which is represented as EA? Okay. So the energy given to reactant, okay, the energy that is given to reactants, to reach at maximum energy stage, and that energy stage is called as transition state. Got it? So that energy level is called as my transition state. So whenever I'm providing the energy to reactants, these reactants are acquiring all the energy and reaching towards maximum energy stage called as transition state. After this, the reaction becomes too spontaneous. So from this point, the spontaneous reaction occurs, and finally I am going to get my product. Okay, so finally I am going to get my product. So what does enzyme do? If I'm considering a normal reaction without enzyme, then the activation energy is like this much. So this is my activation energy, but with enzyme, the activation energy has been reduced down. So I am requiring So I am requiring very much less energy to attain that maximum energy stage. Got it? So whenever the enzyme is introduced, activation energy is decreased, and the the transition state for any particular reaction occurs occurs quickly. So this particular transition state is acquired very quickly whenever I am introducing enzyme in the system, due to which the product formation, due to which the product formation, the product formation takes place swiftly. Takes place swiftly. So the products are forming at a faster rate whenever I am introducing the enzymes due to reduction in the activation energy. Got it? So enzymes are not actually involving inside any chemical process. They are just reducing down the activation energy so that any reaction, suppose A gives B, it takes time 30 seconds. Alright, no enzymes has been used. But whenever same thing, the A give B, whenever we are introducing C enzyme inside this process, then the time required might be 10 seconds. So here we save 20-second time. Why? Because in this case, the activation energy has been decreased, due to which the production rate of B is increased. Got it? So the production rate of B as a product has been increased due to the reduction of activation energy. So this is the function of enzyme. So now what are the types of enzyme based on structure? So if I am considering a structure of this as my enzyme, okay, so this is nothing but my enzymes. So this enzyme has to be in active form. This should be in active form. All right. So whenever I'm considering an active form of enzyme, then and then only the enzyme is going to show its activity, right? So enzyme without any attachment, without any attachment. Now what attachments? We'll see that. Okay. So enzymes without any attachment is called as apoenzyme. It is called as apoenzyme, or it is called as zymogen, which is nothing but inactive enzyme right now. So this is nothing but my inactive enzyme. And we have to convert this particular enzyme into an active form. So whenever I'm considering my first attachment, which is nothing but here carbon chain, which is extra than that of this protein. All right. So there is an organic chain, extra than a protein, is basically called as prosthetic group. So extra part of organic chain, which is non-proteinaceous, in nature. So that particular chain is called as my prosthetic group, and that is one of the important factor which is attached on the enzyme, due to which the enzyme is going to get its active nature. Alongside, there has to be some site. If suppose I am considering one enzyme which is having this kind of site, okay, so in this particular site there has to be attachment of something. Whenever I'm considering a metallic molecule or a metallic ion is going to get attached. So this metallic ion is called as my cofactor. Alright, this is called as my cofactor. And there are some vitamin derivatives. Okay, and there are some vitamin derivatives. Means the structure which have been formed from vitamin. That vitamin derivative is called as coenzymes. Alright, let's check what is the role of coenzymes and cofactor. As we have seen, the prosthetic group attachment is completely driving the enzyme to become active. However, the enzyme only works when it attaches to the substrate. It's as simple as that. Whatever the substrate, the enzyme is going to convert into a product. That substrate has to get attached to the enzyme first, right? So for this attachment, substrate don't have eye, right? It's not a human being. It's just a chemical molecule. So whenever I'm considering a substrate, the substrate don't have his own eye. So in this particular case, the substrate is carried. It is carried towards enzyme by attaching on coenzyme. So coenzyme is basically a carrying substrate towards enzyme. Got my point? While if I'm considering, even though the substrate is rich towards enzyme, if suppose this is my enzyme and the substrate is like this, so it won't fit properly inside the enzyme, right? So we have to see or change the particular site to this. Then this substrate might enter inside the enzyme. So we have to change a site present inside the enzyme according to the substrate. Correct? So in this particular case, even though the substrate is near enzyme, the active site, or which is called as substrate binding site. Simple. So it is also called as substrate binding site has to be open for substrate, and that opening is carried out. So the opening for that substrate, is carried out by cofactor. Even though that is a tiny metallic ion, binding of that metallic ion will switch the structure of active site present inside the enzyme. Whenever I am considering a three-dimensional structure of enzyme, that three-dimensional structure will change slightly, due to which the opening of active site will occur. Got my point? So the structure of enzyme is completely changed from the triangular active site to a rectangular or square shaped active site. So in this particular case, cofactor helps to change the three-dimensional structure of active site of an enzyme of an enzyme. Is that simple? Whenever I'm considering a cofactor, which is my tiny metallic ion, it can be your Mg2 plus, it can be your Ca2 plus, it can be your manganese 2 plus, or it can be your copper 2 plus. There are multiple, or Zn2 plus. So there are multiple cofactors which are specific for specific enzyme. Alright, they are specifically working for specific enzyme and they are governing or regulating. So this helps to, means it is going to regulate the site of particular enzyme for binding to a substrate. Got it? So let's move on to the next part, that is what are different types of enzyme based on function. All right. Whenever I'm considering a function, so enzyme has its own function. So enzyme has its own function that it is going to convert, acting on a substrate and converting a substrate into my product. So enzyme has its own role just to convert substrate into a product and by reducing the time. All right. The total time taken for a reaction, we have to reduce that particular time whenever I'm introducing enzyme inside the reaction. But what are specifically enzymes doing? So if I'm considering a sucrose, sucrose was made up of, guys, it was made up of glucose plus fructose. Correct. So sucrose was made up of glucose and fructose. But that bond between glucose and fructose was formed by glycosidic bond. What if I told you that I want to obtain glucose and fructose separately from the sucrose? I want to obtain glucose and fructose separately from sucrose. Then what will I have to do? That during glycosidic bond formation, I have removed water molecule. Correct? Are you remembering that? So whenever I was forming glycosidic bond, so during that glycosidic bond formation, I have removed the water molecule. So whenever I am introducing water molecule again, all right, whenever I am introducing water molecule again, so in that presence of water molecule, the enzyme sucrase is going to break down the sucrose into glucose and fructose. Got my point? So there is a case called hydrolysis. So this is a category which uses water to break down a bond. Is that simple? The next case will be of kinase. So this kinase enzyme are quite interesting. So if I'm considering kinase enzyme, whenever I'm going to see kinase, that kinase is going to add phosphate group on the substrate. Whenever I have to add phosphate group on a substrate, I will use the kinase enzyme, and those kinase will be specific for specific substrate. So according to the substrate, kinases are going to get changed. So if I can add phosphate group, I can remove the phosphate group as such, right? So during that, there is going to be the enzyme called as phosphatase. Whenever there is ase, mostly it is removal as per your book suggests. But that is not true, as even the ase is there inside the kinase as well. So whenever I am considering phosphatase, the enzyme removes removes phosphate group from substrate. From substrate. As per this category concern, we can differentiate different enzymes based on their functional activity inside the biological system. Got it? So let's move to the next part, that is nomenclature of enzyme. So what is nomenclature? Alright, what is nomenclature? Actually, the nomenclature that comes in your mind whenever the enzymes are there, so they are divided based on EC number. Alright, it is completely dependent on EC number, and that EC number contains class, subclass, sub-subclass, and enzyme serial number. Alright, so whenever the enzyme has to represent, suppose this is my enzyme and it is represented by 2.7.1.1. So there is going to be the enzyme belongs to class 2, subclass 7, sub-subclass 1, and the serial number is 1. That is what the enzyme is going to be suggest, and that is nothing but my hexokinase. Alright, as the example is suggesting here. So whenever I am considering that the classes of enzyme, the classes are completely dependent. They are completely dependent on functional activity of enzyme. On functional activity of enzyme. That is what we have seen currently in the previous slide when we were discussing what are the different type of enzyme based on function. So these are basically categorized into classes. Got my point? So there are total 6 classes: O, T, H, L, I, L. You have to remember this particular sequence as it is: OTH, LI, L. OTH little, you can remember like that. So O stand for oxidoreductase. O stand for oxidoreductase. Now what is oxidoreductase and all? We are going to cover everything in detail in the next video. Alright. T is nothing but my transferase. Then H is nothing but hydrolases. We have seen that case, right? And similarly, if I am considering L, that is lyses, isomerase, and ligases. So these are nothing but the class of enzymes. So the first number is going to represent the functional activity of enzyme. So the first number decides functional activity of enzyme. It's kind of tricky and it's not that useful for the NEET. Alright, it's just for the concept clearance that inside a book, the nomenclature of enzymes is completely different. It is based on the function, and they are just saying that whenever there is A's, they can break down the particular substance, but that is not at all true, guys. Alright, so they are not always going to break down the substrate, but there are some different cases, different reactions taking place inside the whole biological system, which are governed by enzymes. So if I am moving to the book part, so according to your book, there is going to be whenever the suffix ase is used, then that is going to be breakdown of substrate. The enzyme that is going to break down a particular substrate. So for example, if I am considering lactose, so can you guess name of substrate? That is lactase. If suppose maltose, the enzyme for maltose to break down is nothing but maltase. If suppose DNA, there is going to be DNase. Likewise, whenever I am going to add ase at the end, that is going to break down a particular substrate. But is that true? Whenever I am considering hexokinase, then there is ase, but this belongs to your transferase group. This belongs to your transferase group, and as we have seen, kinase is nothing but it is going to add phosphate group. All right, there is one more example called as alkaline phosphatase. It is a phosphatase enzyme itself, but it works in the alkaline condition, that means in pH above 7. Got it? So this phosphatase helps to remove phosphate group from substrate, and this is one of the important enzyme in genetic engineering, engineering. Got it? So this is one of the important enzyme inside the genetic engineering. So this was your introduction to enzymes and what are enzymes made up of. All right. So based on this topic, we are taking from this basic part of enzyme till the advanced one with some of the graphs, how enzymes are going to work, what are the different models for enzyme, and finally how this enzyme work at different conditions. Got it? So for now, that's it. Hope you are loving it. Please share it within your friends. Please do not forget to like and subscribe to my video, guys. Bye bye. See you in the next video.